Dear students, myself Dr. Zubari Arham Javed, lecturer in Botany Department of Microbiology, and today we will discuss about use of physical methods in microbial growth control, in which we will discuss heat control, means dry heat and moist heat. This topic is one of the subtitle of your course outline, control of microorganisms. First of all, we will discuss about our contents of today's lecture. In the beginning, we discuss purpose of microbial growth control, and then here is graphical chart of controlling microbes. Then physical methods in controlling microbial growth, in which we will discuss moist heat, dry heat, moist heat and dry heat with some different techniques like autoclave and pasteurization in moist heat, and hot air oven and dry heat. So let's start our today's lecture. As ever since microbes were shown to cause diseases, as you all know, people have invented different techniques to control their spread, and controlling microbial growth is important in the medical field and also important in the pharmaceutical and biotechnology industries, academic research and food industries. Each antimicrobial substance or agent achieves a different level of microbial elimination by a certain mechanism. As explained in the discussion of growth curves, both the growth and death of microorganisms occur at logarithmic rate. Here we are concerned with just the death rate and the effects on it of antimicrobial agents. What is the antimicrobial agents? These are the substances that kill microbes or inhibit their growth. When we start our today's topic, we must know about the purpose, that what is the purpose of controlling the microbial growth. The purpose of controlling microbial growth is to stop the spreading the diseases or the food spoilage. Here is a graphical presentation of microbial controls method, in which you see these physical agents are divided further into heat and radiation mechanisms or methods, in which you see that heat is further subcategorized in dry heat and moist heat methods. And in dry heat we will study about incineration and dry oven, and in moist heat we will study about steam under pressure, simply means autoclave, and also the boiling water, hot water, pasteurization. And in radiation category we will discuss about ionization and non-ionization. In ionization, here we have X-rays, cathode and gamma rays, and in non-ionizing radiation we will study UV rays. Then class, you also see about chemical agents. Chemical agents are divided into two categories, gases and second one is the liquids. Liquids are also further subcategorized in animate and inanimate category. Animate mean those liquids which are used on the living, tissues. And inanimate means those liquids which are used to sterilize the non-living elements or non-living substances, like our our floor, our ceilings, our windows. Then here are some mechanical removal methods in which we will study filtration, which is further divided into two classes, means air filtration or the liquid filtration. First of all, we study the use of physical methods in controlling microbial growth. And today we study the heat, which is further divided into moist heat, dry heat and low temperatures. Class, today we will cover our two portions, the moist heat, in which we will discuss autoclave and pasteurization, and the other portion is dry heat, which include hot air oven. The third one, the low temperatures, in which freezing, refrigeration and freeze drying. We will cover this topic in our next lecture. First of all, moist heat. As fire and boiling water have been used for sterilization and disinfection since the time of the Greeks, and heating is still one of the most popular ways to destroy microorganisms. Either moist or dry heat may be applied, but when we discuss moist heat readily kills viruses, bacteria and fungi, as you all see in this table. Moist heat is thought to kill by degrading nucleic acids and by denaturing enzymes and other essential proteins. It may also disrupt cell membranes. Exposure to boiling water for 10 minutes is sufficient to destroy vegetative cells and eukaryotic spores as well. But unfortunately, the temperature of boiling water means in the range of 100 degrees centigrade or 212 Fahrenheit at sea level is not high enough to destroy bacterial endospores, which may survive hours of boiling. Therefore, boiling can be used for disinfection of drinking water and objects not harmed by water, but boiling does not sterilize. As you all see in this table, which shows the approximate conditions for moist heat killing, that yeast needs 5 minutes at 50 to 60 degrees centigrade for their vegetative cells. However, if we want to kill their spores or the spores of the yeast, they need 5 minutes at 70 to 80 degrees centigrade. And the same situations with the molds, bacteria and viruses. Then, first of all, we will discuss autoclave. In order to destroy bacterial endospores, moist heat sterilization must be carried out at temperatures above 100 degrees centigrade, and this requires the use of saturated steam under pressure. Steam sterilization is carried out with an autoclave, a device somewhat like a fancy pressure cooker. For generating a saturated steam under pressure, the autoclave is one of the most common and easier and most less expensive instrument which we have to operate in our laboratories. The development of the autoclave by Chamberlain in 1884 tremendously stimulated the growth of microbiology. Here are some specifications, like while using the autoclave, the temperature should be at 121 degrees centigrade pressure has to be 15 psi, and duration will be 15 to 20 minutes. Then if we study about the working, the Chamberlain was the first scientist who studied about the autoclave in 1884. And if we study about the working, then water is boiled to produce steam, which is released through the jacket and into the autoclave's chamber. The air initially present in the chamber is forced out until the chamber is filled with saturated steam and the outlets are closed. Here is the figure about working. From this figure, you can easily sketch an image in your mind that when the air initially enters in the chamber is forced out until the chamber is filled with saturated steam and the outlets are closed. Hot saturated steam continues to enter until the chamber reaches the desired temperature and pressure, usually 121 degrees centigrade and 15 pounds of pressure, as I already told you. At this temperature, saturated steam destroys all the vegetative cells and endospores in a small volume of liquid within 10 to 12 minutes. Treatment is continued for at least 15 minutes to provide a margin of safety. And for larger containers, of course, for larger containers of liquids such as flasks and carboys, there is required much longer treatment times. Here you have figures about countertop autoclave. These are easier to work and are commonly available in our laboratories. This shows a large automatic hospital autoclave. Then checking for sterility. Bacterial endospores will be killed only if they are kept at 121 degrees centigrade, as we all know. At 121 degrees centigrade and for 10 to 12 minutes. But when a large volume of liquid must be sterilized, an extended sterilization time is needed because it takes longer time for the center of the liquid to reach 121 degrees centigrade. As for example, 5 liters of liquid may require about 70 minutes. Therefore, we have some steps for checking the sterility, that either our specimen or our substance is sterile or not. In view of these potential difficulties, a biological indicator is often autoclaved along with other material. Therefore, we have culture tube containing a sterile ampule of medium and also special tape or the paper indicator strip. This indicator, if we study about a culture tube containing a sterile ampule of medium, this indicator commonly consists of culture tube and a paper strip covered with spores of geobacillus stereothermobacillus. After autoclaving, the ampule is aseptically broken and the culture incubated for several days. If the test bacterium does not grow in the medium, the sterilization run has been successful. And second one is the special tape or the paper indicator strip. These that changes color upon sufficient heating is autoclaved with a load of material. If the word appears on the tape or if the color changes after autoclaving, the material is supposed to be sterile. These approaches are convenient and save time but are not as reliable as the use of the bacterial endospores. Here we have graphical explanation of sterility confirmation. As you all see that to check if an autoclave is operating properly, a commercial prepared spore test ampoule is placed in the autoclave and run with the test of the load. Afterward, the vial is crushed to release medium onto the strap containing spores. If the load was truly sterilized, the spores will have been killed and growth will not occur. In the medium, sometimes an incubator dye is added to the medium, which will turn color if the microbial growth occurs due to the accumulation of acid byproducts. This is faster than waiting for sufficient growth to turn the medium cloudy. The second one is pasteurization. Many substances such as milk are treated with controlled heating at temperatures well below boiling, a process known as pasteurization in honor of its developer, whose name is Louis Pasteur. In the 1860s, the French wine industry was plagued by the problem of wine spoilage, which made wine storage and shipping difficult. Pasteur examined spoiled wine under the microscope and detected microorganisms that looked like the bacteria responsible for lactic acid and acetic acid fermentations. He then discovered that a brief heating at 55 to 60 degrees centigrade would destroy these microorganisms and preserve wine for long periods. Milk pasteurization was introduced into the United States in 1889. Like milk, beer, and many other beverages are now pasteurized. Pasteurization does not sterilize a beverage only, but it does kill any pathogens present and drastically slows spoilage by reducing the level of non-pathogenic spoilage microorganisms. Here are some specifications that while working according to pasteurization, we should have temperature around 63 degrees centigrade and duration will be 30 minutes. But nowadays it is passed over by heat exchanger which work at temperature around about 72 degrees centigrade and duration is just near about 15 seconds. Our second portion is dry heat sterilization, in which we will study dry hot oven. As we all know that many objects are best sterilized in the absence of water, means by dry heat sterilization. Some items are sterilized by incineration. For instance, inoculating loops which are used routinely in the laboratory can be sterilized in a small benchtop incinerator, as you all see in this figure. Other items are sterilized in an oven at 160 degrees centigrade to 170 degrees centigrade for 2 to 3 hours. Microbial death apparently results from the oxidation of cell constituents and denaturation of proteins. Dry heat is less effective than moist heat. The spores of Clostridium botulinum, the cause of botulism, are killed in 5 minutes at 121 degrees centigrade. by moist heat, but only after 2 hours at 160 degrees centigrade with dry heat. The food processing industry makes extensive use of thermal death point. After a food has been canned, it must be heated to eliminate the risk of botulism arising from Clostridium botulinum spores. Heat treatment is carried out long enough to reduce a population of botulinum spores. Thus, there is a very small chance of any can having a viable spores. When we discuss about the advantages of dry heat, we have two major advantages, like it doesn't corrode glassware and metal instruments as moist heat does, and it can be used to sterilize powders, oils, and similar items. Most laboratories sterilize glassware and peppers with dry heat. But with advantages, it might contain disadvantages like dry heat sterilization is slow and not suitable for heat sensitive materials like many plastics and rubber items. If we study about thermal death point, then what is thermal death point? Thermal death point is the lowest temperature at which a microbial suspension is killed in just 10 minutes. Dear class, because heat is so useful in controlling microorganisms, it is essential to have a precise measure of the heat killing efficiency. Initially, effectiveness was expressed in terms of thermal death point, which is the lowest temperature at which a microbial suspension is killed in 10 minutes. However, such destruction is logarithmic and it is theoretically not possible to completely destroy microorganisms in a sample, even with extended heating. As you all see here, here is dry heat incineration instrument. So class, it's all about your today's lecture in which we were studying about the dry heat and moist heat. Our next portion, which will be about low temperatures, we will discuss about these low temperatures procedures or methodologies in our next lecture. Here are some references from which today's lecture is prepared. Class, if you have any query relevant to today's lecture, then you freely ask me in Google Classroom. Thank you so much.